Hello dear students. Today our topic of deliberation is chemical methods of food preservation. The main objectives of this lecture are basics of food preservation, criteria to choose food preservation methods, need of food preservation methods, categories of food preservation methods, and the chemical methods of food preservation. Food is indispensable for humans. The supply of food and other essential continues to have a huge impact on the development of human society as a whole. Increasing world population not only causes an increase in the food demand and supply, but also makes the minimization of food wastage due to spoilage imperative. While food production is a significant part of the process to ensure constant, varied, harmless food supplies to cater to the consumer demands, food preservation becomes important to achieve the balance between the supply and demand of food. Following harvest, slaughter, or manufacture, all food items tend to lose quality and texture over time. This loss of quality of foods depends on a number of factors, which includes the type of food, its chemical composition, the type of packaging employed, and the storage conditions the foods were subjected to. All the food items have a tendency to be spoiled at any of the phases, ranging from acquirement of raw materials to the consumption of the food. This tendency of spoilage can be augmented or lessened at any of these stages. This makes the complete preservation of foods a multi-factor process, as it depends on more than one component and requires regulation at more than one of those components to achieve the required results. Now let's move on to food preservation. Food preservation is an action or method designed to maintain foods at a desired level of quality. A number of new preservation techniques are being developed to satisfy current demands of economic preservation and consumer satisfaction in safety, nutritional, and sensory aspects. The strategy for the preservation of foods requires the implementation of some steps whereby foodstuffs undergo some processing. Preservation techniques are numerous and generally differ from one food product or food class to another. In practice, a combination of techniques is sometimes preferred in order to achieve the desired preservation without tampering with the safety or the integrity of the food product. Moving on, this leads us to the basis of food preservation. Preservation is based predominantly on delaying or preventing the growth of microorganisms. It, therefore, must operate through those factors that most effectively influence microbial growth and survival. These include a number of physical and chemical factors and also microbiological factors, which depend on the nature of the microorganisms that are present. The factors have been classified in a number of ways, but the most widely quoted categorization separates the major factors into 5 different types. To begin with, intrinsic factors. Intrinsic factors include chemical and physical factors that are normally within the structure of the food and with which a contaminating microorganism is therefore inextricably in contact. Another type of factors are the processing factors. These, as the name implies, are deliberately applied to the foods in order to achieve improved preservation. Extrinsic factors. Extrinsic factors are those factors that influence microorganisms in foods but which are applied from outside the food and act during storage. Next are the implicit factors. These include those factors that are related to the nature of the microorganism themselves and to the interaction between them and with the environment with which they are in contact during growth. The last kind of factors that affect the food preservation are the net effects. These take into contact the fact that many of the factors strongly influence the effects of each other, so the overall effect of combinations of all factors may not be readily predictable but may usefully act synergetically to give greater effects than one would expect from a single factor. These form the basis of the so-called combinational preservation. Moving on, what causes the basic need of the preservation? The idea of food preservation arises from the desire to maintain the food quality, its physiochemical properties, and maintain the functionality of its nutritious components without affecting the product itself. The main reasons for food preservation are to overcome inappropriate planning in agriculture, produce value-added products, and to provide variation in diet. Throughout civilization, there has always been a need to preserve foods for future use for a number of reasons. These include to maintain the integrity of food products, for increasing the shelf life of food products, for replenishment of food supply during famine, that's the food security aspect, also for transporting of food products for use in a different locality, providing value-added food products that can give better quality foods in terms of improved nutritional, functional, convenience, and sensory properties, meeting the consumer demand for healthy and more convenient foods, allowing for variation in the diet, particularly in underdeveloped countries, to reduce reliance on a specific type of grain, that is rice or wheat. Moving on, the food preservation methods. The preservation and processing of food is not as simple or straightforward as it was in the past. It's now progressing from an art to a highly interdisciplinary science. A number of new preservation techniques are being developed to satisfy the current demands of economic preservation and consumer satisfaction and environment safety. The preservation methods are mainly based on the types of food that need to be prepared or formulated. The factors that are considered before selecting a preservation process include the desired quality of the product, the economics of the process, the environmental impact of the methods. At present, different methods of food preservation are available for food industry. Based on the mode of action, the major food preservation techniques can be categorized into three different classes: slow down or inhibit chemical deterioration and microbial growth. The other classes are those which directly inactivate bacteria, yeast, molds, or enzymes. And the third class are the ones which avoid recontamination before and after processing. Modern preservation methods are designed not only to extend the shelf life of the food, but also to ensure its safety by inactivating pathogenic microorganisms and viruses of concern, or in some cases, just preventing their growth in the product. Moving on, let's look into some chemicals which are used in food preservation. The use of chemicals to prevent or delay the spoilage of foods derives in part from the fact that such compounds are used with great success in the treatment of diseases of humans, animals, and plants. Although a large number of chemicals have been described that show potential as food preservatives, only a relatively small number are allowed in the food products, due in large part to the strict rules of safety adhered to by the Food and Drug Administration, and to a lesser extent to the fact that not all compounds that show antimicrobial activity in vitro do so when added to certain foods. Quite a few chemical preservatives are generally recognized as safe, abbreviated as GRAS. Let's now take a look at some of those compounds most widely used, their modes of action, and the types of food in which they are used. To begin with, one of the important chemicals is the benzoic acid. Sodium benzoate was the first chemical preservative permitted in the foods by the FDA, and it continues in wide use today in a large number of foods. Its common approved derivatives are methylparaben, methyl P hydroxybenzoate, propylparaben, propyl P hydroxybenzoate, heptylparaben, and heptyl P hydroxybenzoate. It has been found that benzoic acid blocks the oxidation of glucose and pyruvate at the acetate level in Krebs cycle. The antimicrobial activity of benzoate resides in the undissociated molecule and is related to the pH. The undissociated form is essential to the antimicrobial activity of benzoate. In this state, these compounds are soluble in the cell membrane and act apparently as proton ionophores. As such, they facilitate proton leakage into the cells and thereby increase energy output of cells to maintain their usual internal pH. The pK of benzoate is 4.20, and at a pH of 4.0, 60% of the compound is undissociated, whereas at a pH of 6.0, only 1.5% is undissociated. This results in the restriction of high acid products such as apple cider, soft drinks, tomato ketchup, and salad dressings by benzoic acid and its sodium salts. High acidity alone is generally sufficient to prevent growth of bacteria in these foods, but not that of certain molds and yeasts. As used in acidic foods, benzoate acts essentially as a mold and yeast inhibitor against yeasts and molds at around pH of 5 to 6 from 100 to 500 ppm are effective in inhibiting the former, whereas for the latter from 30 to 300 ppm are inhibitory. Next important and related chemical compound are the parabens. The common parabens that are permissible in foods are heptyl, methyl, propyl, butyl, and ethyl parabens. In general, gram-positive bacteria are more susceptible than gram-negatives to the parabens. The parabens appear to be more effective against molds than against yeasts. As esters of p-hydroxybenzoic acid, they differ from benzoate in their antimicrobial activity in being less sensitive to pH. The pK of these compounds is around 8.47 and their antimicrobial activity is not increased to the same degree as for benzoate with the lowering of pH as noted. They have been reported to be effective at pH values up to 8.0. Heptylparaben is quite effective against microorganisms at a concentration of 10 to 100 ppm, affecting complete inhibition of some gram-positive and gram-negative bacteria. It's also effective against the malolactic bacteria. Propylparaben is more effective than methylparaben with up to 1000 ppm of the former and 1000 to 4000 ppm of the latter needed for bacterial inhibition. Another important chemical used in preservation of foods is the sorbic acid. Sorbic acid is employed as a food preservative, usually as the calcium, sodium, or potassium salt. Sorbate inhibits microbial cells by the general mechanism involving the proton motive force. It causes weakening of the transmembrane gradient such that the amino acid transport is affected adversely. Like sodium benzoate, they are more effective in acid foods than in neutral foods and tend to be on par with benzoate as fungal inhibitors. Sorbic acid works best below a pH of 6.0 and is generally ineffective above pH 6.5. These compounds are more effective than sodium benzoate between pH 4 and 6. At pH values of 3 and below, the sorbates are slightly more effective than the propionates but about the same as sodium benzoate. The pK of sorbate is 4.8 and at a pH of 4.0, 86% of the compound is undissociated, whereas at a pH of 6, only 6% is undissociated. The sorbates are primarily effective against molds and yeasts, but research has shown them to be effective against a wide range of bacteria. Shelf life extensions have been obtained by the use of sorbates on fresh poultry, vacuum packaged poultry products, fresh fish, and perishable fruits. The sorbates have been studied by a large number of groups for use in meat products in combination with nitrites. The combination of sorbate plus reduced nitrite has been shown to be effective in a variety of cured meat products, not only against Clostridium botulinum but other bacteria such as S. aureus. The widest use of sorbates is as fungistats in products such as cheeses, bakery, fruit juices, beverages, salad dressings, and the like. In the case of molds, the inhibition may be due to the inhibition of the dehydrogenase enzyme system. Against germinating endospores, sorbate prevents the outgrowth of vegetative cells. Moving on, the propionates. Propionic acid is a 3-carbon organic acid with the structure CH3CH2COOH. The acid and its calcium and sodium salts are permitted in breads, cakes, certain cheese, and other foods, primarily as a mold inhibitor. The tendency of propionic acid and its salts towards dissociation is very low, and they are consequently active in low acid foods. They tend to be highly specific against molds, with the inhibitory action being primarily fungistatic rather than fungicidal. With respect to the antimicrobial mode of action of propionates, they act in a manner similar to that of benzoate and sorbate. The pK of propionate is 4.87 and at a pH of 4, 88% of the compound is undissociated, whereas at a pH of 6, only 6.7 remains undissociated. The undissociated molecule of this lipophilic acid is necessary for its antimicrobial action. The mode of action of propionic acid is similar to that of benzoic acid. Another important chemical is the sulfur dioxide. Sulfur dioxide and the sodium and potassium salts of sulfide, bisulfite, metabisulfite all appear to act similarly. Sulfur dioxide has been used as a food preservative since ancient times. It's used in its gaseous or liquid form on dried fruits, in lemon juice, molasses, wine, fruit juices, and others. Sulfur dioxide is bacteriostatic at low pH and concentration of 100 to 200 ppm, as well as bactericidal at the concentrations more than 200 ppm. Molds can be controlled on grapes by periodic gassing with sulfur dioxide, and bisulfite can be used to destroy aflatoxins. Although the actual mechanism of sulfur dioxide is not known, several possibilities have been suggested. It has been proposed that undissociated sulfurous acid or molecular sulfur dioxide is responsible for the antimicrobial activity. Its greater effectiveness at low pH tends to support this argument. Sulfur dioxide is also thought to be an enzyme poison, inhibiting growth of microorganisms by inhibiting essential enzymes. Because the sulfides are known to act on disulfide bonds, it may be presumed that certain essential enzymes are affected and that inhibition ensues. Its use in the drying of foods to inhibit enzymatic browning is based on this very assumption. Moving on to a very common household chemical, that's the salts and the sugars. These compounds are grouped together because of the similarity in their modes of action in preserving food. NaCl has been employed as food preservative since ancient times. The inhibitory effect of salts are not dependent on pH, as are of some other chemical preservatives. The early food uses of salt were for the purpose of preserving meats. This use is based on the fact that at high concentration, salt exerts a drying effect on both food and the microorganisms. When high concentrations of salt are added to fresh meats for the purpose of preservation, both the microbial cells and those of the meat undergo plasmolysis, that is the shrinkage, resulting in the drying of meat as well as inhibition or death of microbial cells. Enough salt must be used to affect hypertonic conditions. The higher the concentration of salt, the greater is the preservative and drying effect. Sugars such as sucrose exert their preservating effect in essentially the same manner as salt. High concentrations of sugar make the water unavailable to microorganisms. One of the main differences is in the relative concentrations. It generally requires about 6 times more sucrose than NaCl to effect the same degree of inhibition. The most common uses of sugars as preserving agents are in the making of fruit preserves, candies, condensed milk, etc. Microorganisms differ in their response to hypotonic concentrations of sugars, with yeast and molds being less susceptible than bacteria. Some yeasts and molds can grow in presence of as much as 60% sucrose, whereas most bacteria are inhibited by much lower concentrations. Moving on to the organic acids. The antimicrobial effects of the organic acids are due to both the depression of pH below the growth range and the metabolic inhibition of the undissociated acid molecules. Organic acids are employed to wash and sanitize animal carcasses after slaughter to reduce their carriage of pathogens and to increase the product's shelf life. Acetic acid and lactic acid are among the most widely employed organic acids as preservatives. In most instances, their presence in the subject foods is due to their production within the food by lactic acid bacteria. Products such as pickles and fermented milks, among others, are created by the fermentative activities of various lactic acid bacteria which produce acetic, lactic, and other acids. The bactericidal effect of acetic acid can be demonstrated by its action on certain pathogens which, when exposed to 10% acetic acid, were undetectable after 10 minutes. Moving on to the antifungal agents used for fruits. A number of chemical agents are employed to control fungal spoilage of fresh fruits. For example, benomyl is applied uniformly over the entire surface of fruits. It can penetrate the surface of some vegetables and is used worldwide to control crown rot and anthracnose of bananas. Both benomyl and thiabendazole are effective in controlling dry rot caused by Fusarium species. To prevent the spread of botrytis from grape to grape, sulfur dioxide is employed for long-term storage. It is applied shortly after harvest and about once a week thereafter. Another important class of compounds are the ethylene and propylene oxides. The ethylene and propylene oxides exist as gases and are employed as fumigants in the food industry. The oxides have a similar mechanism of action, are applied to dried fruits, nuts, and spices, primarily as antifungal compounds. Ethylene oxide is an alkylating agent. Ethylene oxide appears to affect endospores by alkylation of guanine and adenine components of the spore DNA. It's also used as a gaseous stirlant for the flexible and semi-rigid containers for packaging aseptically processed food. Towards the end, let's talk about other chemical preservatives. For example, there is sodium diacetate, which is a derivative of acetic acid. It's used in bread and cakes to prevent moldiness. Organic acids such as citric acid exert a preserving effect on foods such as soft drinks. Hydrogen peroxide is another chemical that has received limited use as a food preservative. In combination with heat, it has been used in milk pasteurization and sugar processing, but its widest use is as a sterilant for food contact surfaces of olefin polymers and the polyethylene in aseptic packaging systems. Ethanol is the common alcohol and is present in flavoring extracts and affects preservation by virtue of its desiccant and denaturant properties. Ethanol vapors produced by a vapor generator can be produced within the headspace of a package, and the vapors have been shown to be effective against some bacteria and fungi. Dihydroacetic acid is used to preserve squash. Diethyl pyrocarbonate has been used in bottled wines and soft drinks as a yeast inhibitor. It decomposes to form ethanol and carbon dioxide by either hydrolysis or alcoholysis. Dimethyl dicarbonate is used as a yeast inhibitor in wine and some fruit drinks at a level of 0.025%. Upon hydrolysis, it yields methanol and carbon dioxide. DMDC has been found to be more effective than sodium bisulfite and sodium benzoate as an inhibitor of E. coli. Dear learners, this was all about today's program. Hope you have learned some new exciting things. Thank you for all your attention. Hope you have a great day ahead. Goodbye.